So, thank you and welcome to the session. I am honored to be co-hosting this session of the International IVF Initiative. For videos and previous sessions, please go to the ivfmeeting.com website and don't forget to leave a review. If you are participating in questions this evening, and we hope you will all do so, please use the Q&A section on the bottom, not the chat function. So very important, put your questions in the Q&A section. My name is Kelly Tilleman, and it's a great honor to be co-moderating this session with Dr. Sara Martins da Silva, who is a senior lecturer in reproductive medicine at the University of Dundee. She runs clinics specifically for male infertility, as well as a sperm studies research clinic for couples affected by unexplained low and no fertilization following IVF ICSI. And everybody, this lady has been recognized as the most inspiring and influential woman around the world by BBC 100 Women. If you want to know more about Sarah, please visit the website.
Oh, wow. Thank you, Kelly. Well, I am delighted to be hosting this session with you. For any of you that don't know Dr. Tilleman, she's currently the director of Ghent University IVF Lab. She's also coordinator of the ESHRA Safety and Quality in ART Special Interest Group. She's Belgium's representative for the EDQM, the European Directorate for Quality of Medicines, and a board member of the international organization ICBAR. Kelly's research experience started in the field of proteomics. She obtained her PhD in 2007 researching biomarkers for arthritis and joined Ghent University Women's Clinic in 2009 after postdoctoral collaboration with Petra de Sutter. Kelly combines quality management with project and laboratory management, and you can see more of her impressive bio on the website. So, tonight's session is entitled Beauty is in the Eye of the Beholder, Human Sperm Morphology Assessment, and is sponsored by Zymote. Now, we're going to be talking about semen analysis and sperm morphology. You will all be aware that the long-awaited 6th edition of the WHO Laboratory Manual for Examination and Processing of Human Semen is currently open for public consultation. So perhaps more than ever, we really need clarity. Tonight, we have a great session planned with some really fabulous speakers.
Yes, thank you, Sarah. And I have the honor, the great honor of introducing our first speaker, Dr. David Mortimer. I don't think actually David needs a lot of introduction, but still, born originally from the UK, did his PhD in Edinburgh, was scientific director of Sydney IVF. His research has been focusing on human sperm pathophysiology. And David has given many, many lectures and have taught many people in workshops and courses all around the world. We have probably all 560 already of us registered here, probably seen, heard, or met David, probably when he was auditing your center as a quality manager auditor. He became a full-time consultant in '99 with his wife, Dr. Sharon Mortimer, and we all know him, of course, from his consultancy field work. So if you need something else from David, please have a look at his bio on our website. David, please.
Thank you very much. Thank you very much indeed, Kelly. Screen sharing is okay, is it, guys?
Yep.
Good, just checking. So, welcome everybody to this the 60th edition of I3, and thank you also to Zymote as the sponsor of this session. So, I'm going to start the session off by giving an overview and some interesting perspectives on, or I hope are interesting perspectives on sperm form and function. This has been sort of the underlying basis of my career since I was actually an undergraduate with my first ever undergraduate research project. So I'm going to get started. Conflict of interest, as far as I'm concerned, I have none for this. This presentation has no commercial or financial interests involved in it, purely academic and educational. So learning objectives number one. first you need to remember is that this is not what sperm actually look like. These cartoons may be cute and illustrative, but they are not what sperm look like. And it's not surprising that so many people have issues trying to assess sperm morphology when these images are blasted at them from all sorts of websites and TV and media, whatever. You need to understand that this is not what sperm look like, and really need to think how they are put together. For most people, it's looking at with the light microscope and a standard Papa-Nicolau preparation on the top left here, and some high power PAP pictures down here. This sort of imaging is sort of the normal way we look at sperm in the andrology lab. And when we do the assessments, we're using these sorts of types of definitions of different types of defects. There's obviously a very, very large range of potential defects, especially as defects typically occur in combination. So the important thing is that when you say you're looking at a normal sperm, well, we don't know it's normal actually. So in fact, the correct terminology nowadays is ideal. Ideal in terms of this is what the ideal human sperm should look like. And that normal looking sperm or ideal looking sperm is really only reflecting the gross normality of spermiogenesis. And while the appearance of the sperm under the light microscope for an ideal sperm likely relates to functional potential, it does not equate to the sperm's intrinsic or innate fertilizing ability or to the man's fertility. For many years, Papa-Nicolau has been the preferred staining method for human sperm morphology because it makes it very easy to differentiate between cytoplasm, here in the green, and an abnormal midpiece, here with the red staining. And for this reason, we still prefer to use Papa-Nicolau whenever possible. But there are many other stains around, and you can look at Diff-Quick when everything looks different shades of purple, Sperm Blue with blue stains. Spermac is an interesting stain, but again, you still can't really differentiate what's happening in the midpiece. Giemsa is not a good idea. H&Es actually can be remarkably quite good, but again, you can't differentiate cytoplasm or midpiece material easily. And Shore is very common in some countries, particularly France. And again, now it's shades of blue. So there's a good reason for using polychromatic stains like Papa-Nicolau. Spermiogenesis, as I'm sure everybody knows, is the differentiation of a round spermatid into what becomes what's called a testicular spermatozoon. This is what gets released from the seminiferous epithelium. So the human sperm is probably the most highly differentiated cell in the body. And when we look at the ultrastructure of the sperm, there is a lot of very precisely located structural components in a sperm cell, in the head region, the neck region, in the midpiece, where you've got the mitochondria wrapped around in like a double helix of sausages. We've got the outer dense fibers and inside the standard 9+2 axoneme. In the main part of the tail, we have these longitudinal columns and ribs that make up a fibrous sheath. And these structures are all totally important for the function of the sperm. So unless all this stuff is put together in exactly the right way, then a sperm was likely to be dysfunctional. So at the light microscope level, we're only looking at the gross normality of spermiogenesis and hence likely functionality. So we are not establishing that a sperm is normal. And we want to look at ultrastructure in more detail. I published a review in MHR a couple of years ago, and I would recommend people to go there. It's a fairly huge paper and summarizes a lot of the pictures that I've used during my career from some of my favorite other researchers. This is some work actually from part of my PhD, so it's very old. But just to point out that when you're looking at sperm morphology in a Papa-Nicolau stain, and you think this is the acrosome, you're not actually seeing the acrosome. Because if you use Triton, which will remove the outer acrosomal membrane and some of the contents, the sperm still look exactly the same in a Papa-Nicolau stain preparation. If you use Hyamine, which takes off the whole, most of the acrosome, you've now got sperm that still look the same. Or even sodium dodecyl sulfate, a very powerful detergent, which takes the tails off pretty much all the sperm immediately. It takes off the entire acrosome, and the sperm in Papa-Nicolau still look the same. So what you're looking at is the region where the acrosome is located, but you're not actually seeing the acrosome itself. If you want to see the acrosome, for example, as part of an acrosome reaction, you need to use much more detailed methods. In this case, this is gold-labeled phenylglutamine lectin, and it binds to the outer acrosomal membrane, both in the equatorial segment and in the main part of the acrosome cap. And you can see when the plasma membrane is still present, then the gold labeling does not reach the membrane. and it only binds in this very specific region. And you can use this with FITC tagging to find acrosome reactive sperm where they have these nice green belts around the middle of the head, which is actually the residual acrosome, equatorial segment of the acrosome after the reaction has occurred. The one staining method that might be showing the acrosome itself is triple staining. This is a very tricky stain to do well. Richard Chacon was very disgruntled many years ago when I said it was a highly capricious stain, but it is very difficult to do. And in fact, when you're looking in the live cells to establish unreacted and reacted sperm, you're looking for pale pink and white in the acrosomal region. A thing that has been bugging me a lot over the last recent years is this thing about vacuoles and IMSI. There are two types of structures which are not being differentiated. You've got these things called nuclear vacuoles, and it's a pale staining region, usually in the acrosomal region of the sperm. It could be anywhere, and it relates to this empty space inside the chromatin. Now, it's not a vacuum, it's actually contains nucleoplasm, but it's not membrane bound, so it's not a true vacuole, and it's just a space inside the chromatin, and these spaces are present to greater or less degree in almost all human sperm. The other structure is called a crater, and you can see why, because you can see in this picture here, you can see that there's this clear area with a darker border around it. And if you look in transmission electron microscopy, you can see that the plasma membrane is intact, and you've got this big depression into the nucleus. So it looks like someone's taken sperm and stuck their thumb into it. And in this surface replica preparation, you can see in this acrosomal cap region here, we've got this crater. It's clearly got a crater. So the diadem or crater defect has been described in the sperm of several domesticated species as well as man. It affects fertilization and fertility, it affects chromatin condensation, and is associated with sperm DNA damage. And in fact, these are the sperm that are being avoided by IMSI selection of sperm for injection into eggs. So what you're doing is actually avoiding the sperm with craters. The vacuoles are undetermined and certainly lesser significance. So you're actually identifying craters. The other thing I want people to think about is sperm and what they are and what they're not. Sperm are not fish or tadpoles or snakes. To understand the true relationship between sperm and their environment, we need to consider the physics that controls that system. You simply can't extrapolate from your perception of the physical world, because if you were in a swimming pool or whatever swimming, you're in a Reynolds number environment of about 10 to the 4. Fish, it's about 10 to the 2, but for sperm, the Reynolds number is 10 to the -4, so 8 orders of magnitude difference. This means that to sperm, water is not wet, but sticky. And for you to experience the same environment, to have the same R, you would need to be in a pool of molasses. So you can understand stickiness. Inertia is totally relevant at low Reynolds number. So pushing against the water is an invalid concept. It wouldn't actually generate movement, no space gain. And in fact, cyclical motions or deformation of the organism, like what the engineers call reciprocal motion, does not generate movement. Simple whip-like flagellar beating can't generate displacement of the cell. You need asymmetry of the motor effort, and in this case, for sperm, it's a 3D component of the flagellar wave, which creates a helical path. The other thing, of course, is that this low Reynolds number, streamlining is irrelevant because it is not the same as streamlining that we perceive with a Reynolds number of 10 to the 4 or higher if you're putting something in air. So this paper was published a year and a half ago, which says how sperm had people fooled for 350 years. It's a nice paper, but they should have read some older literature, and there's a lot of it around, and they generated a lot of chat on the Sperm Mail bulletin board. Because I was taught when I was a zoology undergraduate in 1974 in Bristol that sperm swim in helical paths. This was first described in 1965. So that was 55 years ago. So we may have fooled us for 295 years. It did it for 350 years. And this paper was a little bit overselling itself, I think. And we've known a lot more about this pattern of movement. This is some information from David Woolley, who in fact did his PhD for Alan Beattie, like I did. And in 1978, he presented a paper that conferring asymmetry to the flagellar wave creates what's called a twisted plane for the flagellar wave as it propagates along the second half of the sperm tail. And this is what causes the sperm to rotate as it swims and causes it to follow an oval-shaped helical three-dimensional trajectory. And in fact, some work done in the lab, I used to do a postdoc in Paris, which is where I met Rolf. They looked at the asymmetric termination of the dense fibers in the tail. And you can see that about 21 microns down the tail, there's an asymmetry appears in the rate of order of termination of these dense fibers. And this is what can create the asymmetry, and will therefore cause the twisted plane and cause the sperm to rotate as they swim and make this three-dimensional helical path. So this is some nice work here, but it's not news. It's been known for quite a while. If you want to understand sperm physiology, you also have to think about what controls sperm behavior and function within their natural environment. And the ejaculate that's collected for semen analysis is actually an artifact. So this is not the natural environment. We're seeing something in a semen analysis where all the fractions are collected together, which never exists in vivo. So in vivo, the sperm are in the prostatic fluid when they meet the cervical mucus. And they're going to get from the site of insemination around the external cervical os up to the site of fertilization in the lower part of the ampulla of the fallopian tube, where they will meet the oocyte. Now, this is a very old drawing, one I drew actually also when I was still doing my PhD. The cervical sperm reservoir is still of uncertain existence, and it does seem that the most likely location of the functional reservoir in humans is in the lowest part of the isthmus, the proximal part, just above the uterotubal junction. And it's incredibly hard to study because it's actually in that part of the isthmus that's embedded inside the uterine wall. But in terms of sperm getting into the tract, at the level of cervical mucus, with semen here and mucus here, sperm start to swim into these little channels that they create called phalanges, and eventually the sperm will colonize the mucus, and the mucus constrain them all to swim in the direction of strain, which is basically upwards. So my work for my PhD thesis was on sperm selection and during sperm transport in humans and rabbits. And my friend Tony Blamley drew this cartoon in the late 1970s. And it's a very good illustration of what you see in terms of sperm transport. In the semen sample here, we've got dead, dying, immature, abnormal, agglutinated sperm. And amongst all these sperm, a few handsome, fast-swimming sperm. And these are the ones that get into the cervical mucus and will reach the upper levels of the female tract. In vitro, you can study the same thing. And this device was also used in part of my PhD thesis, built by a guy called Rodney Kelly. And it's basically a two-chamber compartment with a 11-micron poured nickel mesh or grid-sized nickel mesh separating the semen and the medium, and the sperm just swim up into the medium. And this allows you to separate sperm entirely by motility. And when you look at the sperm here, if you look at the sperm in the seminal population, where they've got a normal-shaped head or an abnormal-shaped head, you can see there are coexisting mid-piece and tail defects in about at least a quarter of the normal head shape, but at least half of the abnormal shaped head. When you select the sperm in terms of their motility by using this sort of device, or even just a straight swim up, this is just an interface stabilized swim up. You can see that the normal sperm have now got, with a normal head shape, have got much fewer mid-piece and tail defects, and the abnormal sperm, there's still some with mid-piece and tail defects, but they've been greatly reduced. So what you see if you select sperm by their motility is a concomitant selection for essentially normal head shape, because those are the ones with fewer mid-piece and tail defects. You can see that quite simply here. This is an unselected sperm population, and we've got some sperm with mid-piece and tail defects shown by the arrows. And if you select them by motility, then you've got no neck, mid-piece or tail defects visible amongst that population. Having recovered sperm from the human oviduct, I can tell you that you will find the occasional sperm with abnormal shaped head, but I never saw any with mid-piece or tail defects in the human oviduct. This selection of bicervical mucus goes back a long way, and this is work that was seminal work when I was doing my PhD, and this whole concept of estrogenic and gestogenic mucus for controlling receptivity of the mucus, followed by Eric Oderblad, was the idea that sperm could not get into gestogenic mucus because it was a very dense meshwork, and when it was estrogenic, you got these nice channels formed by these branching macromolecules. Actually, Udin in Jim Overstreet and David Katz's lab showed, in fact, the mucus is not quite structured like this, but sperm do swim through mucus, but it is very much a selective environment for them. Also, back in the 5 decades ago, people would talk about sperm transmitters of vital mucus, about filtering the sperm by streamlining. And they said this mathematical transformation of a circle into a shape resulting in the least disturbance of streamline would produce a sperm head looking like this, which you might think looks like the human sperm head side on. But Reynolds number says this streamlining is utterly irrelevant. And if you think streamlining is important for sperm biology, then just look at these different mouse sperm. These are Australian old endemic rodent species studied by my old friend Bill Breed. And you can see some of these even look more like inflated rubber gloves than with some sort of streamlined morphology. And these are all perfectly normal sperm for that species, and they're all fertile. So streamlining is not of importance for human sperm morphology in terms of sperm selection in mucus. We also know that abnormal sperm can and do fertilize. This is an example here of a first cleavage mouse embryo, and here there is a diploid male-derived chromosome complement. So this came from a diploid sperm. Diploid sperm exists in rabbits. In fact, my PhD was chasing diploid rabbit sperm at the female reproductive tract and seeing if they got to the site of fertilization. The answer is some of these with one tail will, the ones with two tails don't. Diploid sperm exist in humans, and we could actually, by measuring DNA content, we know that small head sperm are typically hypohaploid. What people call pinhead sperm actually have no heads. They're actually free tails, not sperm. Large head sperm with about a 1.6 times head area like this, they're actually typically diploid by measuring DNA content. And that's why if you find sperm like this and you're trying to do ICSI, you do not go looking for a larger pipette, because these sperm can produce triploid embryos. Our interpretation of perception of sperm morphology has changed a lot over the years. Rolf will look at this in more detail, but over the generations of the WHO manual, the proportion of normal you expect to see has gone down, not necessarily because morphology has changed that much. It has a little bit, but the main thing is because our perceptions of it and how we look at it is different. And this concept of looking at the head, neck, midpiece, and tail was introduced at the 3rd edition of the manual. And at that time, there was concern that at this stage, WHO looked for and counted defects in each region of a sperm that might affect sperm function, normal forms being those with no recognizable defects, while the Tygerberg criteria, strict criteria, defined typical sperm and counted the number that met these criteria. Just after that, Rolf and I did a crossover study. We each produced 60 slides, read them ourselves, sent them to the other person. All results were actually correlated by Lynn Fraser in London, so we never got to see each other's slides. Results where we still had their slides. And in fact, there was very little difference when we considered borderline forms as abnormal. We agreed very, very closely on those assessments. And the problem is that when you have such small proportions of ideal sperm, you've got very few sperm, very narrow dynamic range of results in most patients. You need more information. You need to have some other information about the sperm morphology. This is where things like the acrosome index or teratospermia index come in, because it's telling you other measures of how dysfunctional spermiogenesis was in that man. The other problem is that there are significant issues with counting error and measurement uncertainty, especially if only 100 sperm are counted. If you only got counting 100 sperm, you've got very great statistical sampling variability. In fact, if you want to tell the difference between 3% and 5% normal forms, you actually need to count at least 1,500 sperm, and no one's going to do that. So other information is really very important. So take-home messages. Remember that sperm are single cells. They're not small fish, eels, snakes, or tadpoles. Sperm form and function are tightly integrated. Fertilization depends on normal or functional ultrastructure, as well as fully functional biochemical pathways. Visual sperm perfection by light microscopy, those ideal forms, is not the same as functional competence necessarily. Morphologically abnormal sperm can and do fertilize even in vivo, although there are mechanisms integral to sperm transport to female tract that minimize this possibility. Sperm morphology assessment schemes need to be founded on sperm biology and real-world physics, not on simple human perceptions. We have to avoid anthropomorphism. And then the day, understanding functional morphology, including ultrastructure, is just as important as biochemical pathways and molecular biology. So with that, I will end this part of the presentation, stop the share before something else goes wrong. And
David, thank you so much. That was great. So lots of questions coming in, which is fantastic. I'm going to give you one just now. If morphologically normal sperm can and do fertilize eggs, do we really need strict morphology, particularly if we're performing ICSI?
If you're doing ICSI, yes, because things like those sorry, I've lost you now as well. Now, so you'll be back. There you go, you're back. Sorry, I don't know what's going on today. If you have those craters present, you need to avoid those sperm. So a crater would be a defect picked up by careful morphological assessments. You'd know the prevalence of those things. So morphology is important. I personally don't believe in doing ICSI in everybody. I think it's a very poor approach. There are many other things that happen in terms of sperm biology that are being bypassed and in fertilization biology. So if you want to do sperm morphology assessments, you need to use an assessment scheme that is based on sperm function, not just on prettiness. And I think Rolf will be explaining that quite clearly in the next presentation.
Thank you very much. So leading straight on, next up we have Professor Roelof Menkveld, now retired, but still an associate professor in the Faculty of Medicine and Health Sciences at Stellenbosch University. He has a very illustrious past, previously professor and chief scientist, specialist scientist and director of andrology at Tygerberg Academic Hospital and Stellenbosch University, and played a really important and really long-standing role in the ESHRA Andrology Special Interest Group. Now, one of his most notable achievements, many of you will know, was he is the founder and author of Tygerberg strict criteria for the evaluation of human sperm morphology. Now, that was part of his PhD dissertation in 1987, but you will know, of course, that it is still part of our official WHO 2010, anyway, method of morphology evaluation. So thank you, Rolf.
David asked me to speak on origins, evolution, and clinical significance of the Tygerberg strict criteria. Okay. Okay. So nothing to declare. And the learning objects is just to give an overview of how the sperm morphology evaluation developed over years. The morphology evaluation for animals has been done much longer than for the human. And in the animals, we have the situation that in most domestic animals, we have a very homogeneous sperm morphology picture. So looking at the morphology, it was quite easy to see that the model form, they all more or less look the same. So it was very easy to decide what could be regarded as a morphological normal sperm. However, in the human, the picture is quite different. As human sperm morphology is very homogeneous, heterogeneous, and therefore the approach for the domestic animals could not be followed. So what they did is that they looked at all the different sperm morphology types which they were sure were abnormal, and they were classified and counted, and everything that was left was regarded as normal. So the normal were actually identified by elimination of all the abnormal forms. And then further to add to the problem was that most of these representatives of so-called normal or abnormal were very schematic, which created a sort of false picture. And therefore, this approach to determine normal morphology didn't work for the human. So the very early approach, the theoretical disadvantages is that there was no specific criteria for normal forms it was said if it's not normal, then if it's not abnormal, it must be regarded as normal. And this led that the normal population actually would consist of an abnormal population and also a true normal population. And therefore, if we have the correct normal morphology population, we could expect a good correlation with normal sperm function and normal fertilization and pregnancy rates. So statements that has been appearing in the literature on sperm morphology was inter alia by Menkveld in 1955, who said only most bizarre forms are regarded as abnormal. And Page and Holding in 1951 said there are no criteria for normal morphology, only criteria for abnormal classes. And further, they said that if sperm is borderline or doubtful, it must be regarded as an abnormal sperm. In 1971, Eliasson gave a more descriptive approach where he introduced measurements for a normal sperms which could be considered as being too large or too small or elongated. And he also introduced the multifactorial approach where he said when you do sperm morphology evaluation, you must look at the whole sperm, not at just only at the sperm head. So the whole sperm must be normal to be regarded as a morphological normal sperm. So this method became known as the liberal approach for sperm morphology evaluation. The strict approach, which we developed at Tygerberg Hospital, was conceptualized in the late 1970s, and it was a biological-based concept for normality, based on what we have seen sperm morphology look like after they've been penetrating through good cervical mucus. The sperm selective capacity of cervical mucus for normal sperm has been well documented, for example, by Fredrickson and Borg, also by David in 1982. But as far as I could establish, the concept to use what you see in good cervical mucus has not been used to describe a morphological normal sperm. So when we look at sperm in cervical mucus samples, we can see that we have a homogeneous sperm population pattern with very small biological variations in form, which we will find in any biological system. This concept has also been confirmed by sperm. We have seen bound to the zona pellucida, where there is also a very strong selection for what we then regard as a morphological normal sperm. On the literature of the origin of strict criteria, we first did a lot of work, myself and my colleague, Mr. Frikstander, where we actually did morphology evaluation studies, but unfortunately it never got published. Then I described it in my PhD dissertation, and then also in the well-known 1990 publication which appeared in Human Reproduction. So the principles for evaluation of sperm is that the whole sperm must be evaluated as per Eliasson. The head must have an oval and a smooth form and a smooth contour. The acrosome must be well visible and contains about 40 to 70% of a normal sperm head, and it is identified by very homogeneous light blue staining. Then also to be considered as normal. No other neck or mouthpiece or tail defects may be possible present, and also cytoplasmic droplets or cytoplasmic residues, which is the more common term which is now used, of less than 30% of a normal head. And then the big difference between the previous classification was that we decided that if it's normal, if it's a borderline normal, or we are not sure where to place that, then we will regard this as a morphological abnormal sperm. So the theoretical advantages of this evaluation method was that we expected better repeatable results between and within observers. Also to have a better prognostic value, because we are working with the true morphological normal sperms, which is also a reflection of this morphological sperm function, because you have to have a morphological normal sperm for normal sperm functions to occur. And there was a very good correlation with the pregnancy rates in vivo. The theoretical disadvantage, however, was that due to the stricter application of the criteria for morphological normal sperm, that there was a substantial decrease in the number of morphological normal sperm. So when we look at some studies, we can see that a study done by Uninger in 1988, they found that factors which influenced the sperm, the fertilization, the oocytes were responsible for about 19% for when WH criteria was applied, and 13% in the group where they applied strict criteria, where the oocyte of morphology was abnormal. It was 7.7% with WH criteria and 13% was strict criteria. And then the big difference was seen for normal sperm morphology, where abnormal sperm morphology, where it was WH criteria, only 32% of the sperm could be considered as abnormal, while with strict criteria, about 61% was considered as being abnormal. And this then led to a result where, when you use the WH criteria, there were about 40% of patients which were regarded as so-called unexplained infertility, while with the application of strict criteria, this percentage was brought down to only 11%. When we look at studies where we compare WHO with strict criteria evaluation, there was an article by Bernstein in 1995. The average percentage of normal was strict, with the WH was 21%, and was strict for the same sample was 10%. And then a study which we performed ourselves, we found that with WH criteria, normal percentage was only 31, and with strict criteria, a very low 4.8%. And this is represented in the graph how the average morphology at Tygerberg Hospital changed since 1968 until 2008, from where the low percentage more or less stayed the same over years. So a possible reason for decline in percentage of normal values was the application of strict sperm morphology criteria, Unfortunately, when people started to apply the strict criteria, they thought that strict meant that you have to be very strict in your evaluation, and that the slightest little abnormality should be taken as abnormal, and that was not the intention of strict criteria. It was just the intention that you should have taken a stricter approach, taking the whole sperm into consideration, and where you have seen obvious abnormalities, then to regard that as abnormal. Another factor contributing to the decrease in percentage of normal forms was the negative environment influence. As we all know that we are exposed to, and sperm morphology is one of the most sensitive parameters reflecting any stress on the tested. As we went on with our sperm morphology evaluation, we also recognized additional sperm morphology abnormalities. So abnormalities seen in the neck region, seen at the midpiece, and so on. So we were able to identify additional abnormalities which we thought may add to non-function of the sperm. So this had the consequence that over the years the morphology, normal morphology, became lower and lower and lower, and that was also represented in the consecutive WH manuals, with the final result of the 2010, where they eventually decided that the normal morphology was only 4%, where in the 1987 edition it was at 50%, 1992 at 30%, and in the 1999 manual they actually didn't give any normal cutoff value, but they said that everyone should establish its own values, but when it went below 15%, you could consider that there maybe have been a problem with sperm morphology. So due to the very low percentage of only 4% with the 2010 WH manual, there were many negative reports, and a lot of articles said that doing sperm morphology has no clinical value anymore. And this was also echoed by the Vienna Consensus report. They said unless determination using a more robust methodology, sperm morphology forms should not be considered as for routine evaluation of the infertile couple. So as a consequence, many persons tried to find additional criteria which could sort of evaluate make contributions to find other factors of sperm morphology which make contributions of deciding what may be normal and what is abnormal. And one of these factors was to look at sperm morphology patterns, because in many patients you can have very pronounced patterns which is very specific for a patient. For instance, here they may present with style abnormalities. They may present with abnormalities of the midpiece where the mitochondria has been shifted to the head or to the tail part. There are you may have where a patient where all the sperm are presenting with vacuoles in the acrosomal area of poor staining together with cytoplasmic material. You also get patients where they have predominantly very large sperm, which may be an indication of poor DNA present in the sperm. You have a pattern where all the sperms are elongated, which is called the stress pattern. And you also have patients with mainly very small sperm, a small acrosome, and this is considered as one of a very important factor where, because we know that these patients do very bad with natural IVF or in vitro fertilization. There was an article by Akir taken another look at human sperm morphology, where they reanalyzed the sperm morphology data with regard to where they identified 15 different sperm abnormality categories. And they found that head, mid-piece, and tail defects showed different distributions in fertile males and other group of males, providing a lower and lower threshold in this group of men. But unfortunately, there was such a big overlap that it wasn't really applicable to make a better diagnosis. So we also tried to look at the same principle as Jacquet in a study we did at Fiora. That is an IVF laboratory in St. Gallen in Switzerland. The aim was to investigate if a detailed sperm morphology evaluation may be able to identify a single or combination of specific abnormalities which can improve the prognostic value of sperm morphology. We did a literature study where we tried to find as many abnormalities as possible which have been described in the literature, and eventually we identified 77 different sperm abnormalities. And then we did a retrospective study where we used IUI smears of IUIs performed on patients. The study contained 66 patients, and the outcome was if the patient became pregnant, yes or no. And in the prior results, where we included 36 patients, we had a mean percentage of 2.8% normal forms. Of the highest abnormalities, we found as patients with very large acrosomes, patients with small acrosomes, and one of the most frequent occurring sperm abnormalities were patients with very small heads and also patients with thick mid-pieces. But our conclusion was that we couldn't find any different indications for any specific abnormality, which may be more important in the identification of possible patients with poor morphology, which may contribute to poor fertilization. The disadvantage of the study so far is that it only included a very small number of patients. So really, it's too small to make any definite findings. So then I just want to make a summary that strict criteria has or had a very good predictive value for in vitro fertilization outcome. But due to the decrease in normal sperm morphology values, over years, the strict criteria resulted in loss of prognostic power. Solutions for better diagnosis in the less than 4% abnormal group of the WH manual may be the identification of single or combination of sperm morphology abnormalities, which was not successful. So at the moment, just looking at different kinds of sperm morphology abnormalities, including tail abnormalities and so on, at the moment, there's no single or combination of sperm morphology abnormalities which can help to predict a better prognosis or poor prognosis for the infertile couple. Then I just want to acknowledge the work done at Fiora by Philip Ross and Mrs. Sanderstein. So thank you very much for your attention.
Thank you very much, Dr. Roelof. We have quite some questions in the Q&A, and one was really highlighting, ask him please, please, please. So here is the $1 question for you. Is there any evidence that high level of vacuolation, leads to miscarriage?
I have no idea. Unfortunately, we come back to the question that David has also proposed, that vacuoles, craters, and I would like to ask David that we speak of cysts, but it may be the same, that those patients, because they have problems with the sperm and brain, they don't do good with IVF or normal fertilization. So it's definitely an abnormality which should be taken in consideration. And I see that in the WH manual, they said that more than 20% of vacuoles present, or vacuoles occupying more than 20% of the sperm head, is also a poor prognosis indication, but unfortunately I couldn't find any thing in the literature to sustain this. So if somebody has some more information on that, I would be very grateful that they can present that to us.
Okay. Thank you. So Dr. Ghulam Bahadur from the UK, I hope you have your answer there. Otherwise, we will ask the panel again afterwards. Now we have to move on to our last speaker of today, which is Dr. Lars Bjorndahl. He has a basic medical training at Karolinska, which he finished in the late 70s, early 80s. He did a postgraduate student thesis on human sperm chromatin stability and zinc. In the 90s, he developed an andrology laboratory at Karolinska University, and in 2000, he got recruited by Birmingham's Women's Hospital and the University of Birmingham. Spending 10 years in Birmingham, he moved again to Karolinska, where he is now heading an andrology laboratory and a tissue establishment. Very important for this evening is that Lars is the chief of the revision leading to the 6th edition of the WHO Basic Semen Analysis. So I hope all of you, already 645 people attending this course, have all read the 6th edition. And so you can ask Lars any question that you have on that one. And he is also the project leader of the ISO standard that is coming up for basic semen examination. Lars, please.
Thank you very much for that kind presentation. I should add too that the andrology laboratory at the Karolinska University Hospital was started by my PhD supervisor, Ulrich Christ, and I took it over as head of that when Ulrich retired a few years ago. But it is a very important matter that we are discussing here, and I would like to thank David and Roelof for the excellent presentations. My background is that I was brought up as a physiologist in basic science, then I became a physician, and then a lab specialist in clinical chemistry, and I've been working the last 30 years in trying to implement laboratory quality control matters into laboratory andrology, and that is the basis for what I'm going to talk. And as you will understand, I have learned a lot, not only from Ulrich Quist, but also from David Mortimer, and it will be quite obvious. So I will talk a little about training strategy and about internal quality control and external quality assessment, and then discuss reference limits and decision limits, not the least because we are trying to make that more clear in the revised edition of the WHO manual. So what are the basics in training strategy? Yes, of course, discuss the principles of, for instance, sperm morphology. So every time we got a novice into the laboratory, we go through, explain and show publications from Roelof and David and other pertinent persons. And then we also discuss the principles of the training. And we usually start with common observation. That is, we load a slide into a microscope. We have a computer screen and link to a microscope camera. And then we look at the sperm together and discuss. Of course, you can also use a teaching microscope, but it's very important for a new person coming into this to look through several spermatozoa with a good staining and so on, and discuss what are we looking at and so on. And then we continue with individual training, and that is according to what I always in our laboratory call the Mortimer method. We have training material with known target values, and the novice starts to evaluate them, and after 10 to 15 samples, we compare them with the archive or expert results and give feedback what was good and where the main differences were. And then we can also go back to common observation to discuss if there is a major problem, for instance, with tail assessments. And then the series of material or slides are repeated until the novice comes to very close to zero mean difference from the experts, and then 95 range of variability goes down very much. And this is easier to look at what David presented in the practical laboratory andrology, percentage normal sperm. Now we should say ideal, but we see it's a huge variation in comparison with the expert results in the first set of samples. And then when samples are repeated with feedback in between, we are very close to zero. In my experience, we usually start out somewhere here with a exaggeration of percentage normal, and then when they start to learn about the strict criteria, it goes down to zero. And the ideal is to come close to the plus minus 5% in the 95% variability. So that is a training strategy. And when David talked about this, I thought, yes, but he's quite experienced. So I tried it with several people in our laboratory during the years, and I really see that it works. Sometimes it takes a longer time, depends very much on the individual, but it works. And the important part is to do a good feedback and not only give numbers, but actually sometimes say, this slide was misinterpreted. Let's look at it together. We also have retraining if a member of the staff is way, for instance, maternity or paternity leave for a long time, then we have material, the training material, to see that they rapidly get into the system. And also, if we find in the internal quality control that one individual has repeated or very large deviation from the others, then we can start the retraining. And the good thing is then that we have lots of material with known true values or target values. And this, I should say, is inspired by Susan Rothman in the US, former president of the American Society of Andrology, who talked about the structured way of doing the analysis. And I based this from my long experience of evaluating EQA and seeing how how difficult it is if you just look at the head first and, oh, it's abnormal, which is the most common. And then many people have not been trained properly to actually look at the other parts of the sperm to see if there are also mid-piece defects, tail defects, if there is an abnormal cytoplasmic residue. So therefore, the suggestion, it will be something like this in the coming WHO, a suggestion to start to look at something. Is it a sperm? Well, if it's not a sperm, identify what sort of cell it is and score it separately if required in the situation. But if it's a sperm, can it be classified? Well, then it should be complete with head and tail, should be clearly visible and not overlapped by other cells or objects. If it cannot be classified, stop and go to next cell. This is a oversimplified thing of doing. But the next question, is a large cytoplasmic residue visible? Yes, then you score it as abnormal and score the cytoplasmic residue of abnormality. And then we have to go up here because the format of that, this figure was didn't work into this type of PowerPoint slide. But the next question is the tail defect? Yes, if it's defect, then you score abnormal sperm if you haven't done it already, and tail of normality. Of course, we should say if you had scored a large cytoplasmic residue, you could go down to this question. If you have no abnormal cytoplasmic residue, you go directly to this question. And the system is like that. So you go, so to say, backwards. You start with the cytoplasmic residue, you look at the tail, you look at the mid-piece, and then finally you look at the head. And in my own experience trying this in our laboratory, yes, that is a way to not just so, oh, it's an abnormal head, the sperm is abnormal, and then forget about the rest. So this is the way of structured way of looking at the morphology. It takes some time to start doing it if you have been trained in another way, but after a few slides of a few hundred sperm, it goes as fast, but you don't forget to look at all parts of the sperm. Some words about internal quality control. Well, the basis is still robust laboratory techniques. It's about handling of the samples, making smears, fixation, staining and mounting of smears, and then the training of assessments. And one important part with the methods should be that there are sufficient numbers assessed. And this was a way of looking at the total number of assessed cells and the proportion for the investigated quality. If we look, for instance, for 50% normal tails, if we had looked at only at 100, it's a plus minus 20%. If it's 200, plus minus 14, and the recommended looking at, if you really want to have a low influence of random errors, it's plus minus 10% at looking at 400. If we look at 5%, which is closer to where we find the percentage of normal sperm, We have a similar way. If we look at only 100, it's a huge variability. And David mentioned that if we want to distinguish between 3% and 5%, we need to assess at least 1,500 sperm. David and I made some statistical calculations on that to really understand what do we need if we, and we have to go far away up here. So that is important to know that it's an indication, it's not an exact value. The limits published, not the least by Roelof, is that it's not a limit saying that under that limit all men are infertile, or above it all men are fertile. It's just that probability for fertility is lower when it's under, and it's higher when it's above that. Another thing about internal quality control, we should do comparison between staff members. For each member, compare different occasions, so to see that there's no temporal drift for the individuals, and to see that in a group of staff members, there is no huge variability in each sample. And very important is to do trend analysis for several occasions. And this can be described like this. I took some data we had on morphology, the percentage normal or ideal, calculated a Z-score. And that is maybe a little complicated, but that's a way where you can compare results from different samples with different percentages and so on. But we can see here the first two that were involved in this, they varied a lot. And the more they were trained with this, they were given in the internal quality control, they were given feedback. And after a while, they came much closer to each other, and we should look at this end and this end, and we can see that also this yellow person came close to zero, very close to zero, and they all gather around here. And I would say it's actually the fact of having an instrument where each individual says, oh, I'm always high. Maybe I should change my assessments a little. And I'm always high variation in this. And people want to give a good result. And by doing this repeatedly, and I would say each dot is one sample, and it's a time relation here. So these were the first samples, and these were the most recent ones. So that is very important with internal quality control, not just examining one occasion, but in each point is actually the average of the last 8 measurements. And that means that the average is getting closer and closer to zero variability. It will never be zero, but it's getting closer. So what about external quality assessment? Are results comparable or correct? And that is important. Patients change centers, and if there is if both centers or all centers are participating and performing equally, then results can be changed, can be used from one center, can be used in another center. We don't have to repeat it a number of times. And also that their own centers' use of results from other centers in many different ways. And one important question is it justified to implement procedures from other centers, like reading papers in PubMed? Oh, they are doing like this. But if we are not doing, especially here in this presentation, sperm morphology assessment in the same way, then it can be completely wrong to implement a procedure. And the same thing like reading a paper, picking up things from conferences and workshops and other sources like that. If you know that you are doing the analysis and come to the same results or similar enough results, then it can be okay to implement procedures. But it can be devastating to use procedures from centers that use completely different techniques. and different assessment schemes. So, finally, about this matter with reference limits and decision limits. First, I should mention, and Rolf mentioned that earlier it was presented as normal values, and that is misunderstood in large part, at least in the medical world I've been, being active. Normal values does not point out that the values are normal and anything outside this distribution is abnormal. It's actually the old way of describing a normal distribution, and that nowadays is called the Gaussian distribution, with, or sometimes bell-shaped distribution. So, and then it was ways of saying, well, if it's at these extremes, then it's that is where we make the limits. Now we talk more about reference data, results or data coming from a relevant population. And it should be a population without the disorder in question. And if that should be possible to use, it must be a very limited overlap between results from the reference population and persons with the disorder. And this is an example. If we take the green line here as the reference population, and the red dotted line as the population with the disorder, if it's a very small overlap, then the errors in when we, for instance, have an individual with a result here could be, but with very low probability, a person with the disorder is much higher probability in this area that it's a person with without the disorder. But if we have a huge overlap between reference population and persons with disorder, then a value somewhere here, for instance, if it is, it's a very huge probability, although if we are in the area above a potential limit, we get a very high proportion of the persons with disorders will be over the limit, and a very high proportion of the reference population will be under the limit. So it's very important to know if the reference population is overlapping with the disordered population. And just for clarity, reference values is only the values obtained from a reference from the reference individuals. The reference individual is a person selected based on well-defined criteria, and all the reference individuals make out the reference population, and then we take the reference values from these individuals in the reference population, and we get a distribution of their values. And that distribution contains all values, but that is with outliers and so on. So therefore, the usual thing is that we calculate a reference interval, and that is usually the central 95% of the reference distribution. And we make the reference limits. And the reference limit, that's a value derived from the reference distribution and used only for descriptive purposes. While observed value, that is the value obtained from a test individual and should be compared with the results from the medical population. And I would say the problem with the WHO, it's an enormous material on men in fertile couples. The problem is that we don't have anything of this. We have only this distribution of results from fertile men. And that means that the 95%, central 95% of this distribution does not, or the lower fifth percentile does not constitute a limit between fertile and infertile. And therefore, it's important to bring in this last thing, medical decision limit. A limit based on clinical aspects relevant to the specific situation and not related to the reference limits. And there are a number of such occasions in the, when we talk about examination of the human ejaculates. Something that has been overlooked, I would say, is to find diagnosis of disorders related to the male reproductive organs. We have much attention has been on prognosticizing the result in ART or the spontaneous pregnancy rate and so on, but not so much attention has been to find out malfunction in the male reproductive organs. And there are a number of points where we could look at decision limits instead, not saying that we only examine those with results under the 5th percentile of the fertile men. So, for instance, the choice of further lines of investigation of the man. For instance, when can an endocrine disorder be expected? When would we expect a hypogonadism to be found? And when can genetic disorders be expected? There are different levels or limits than those from the distribution of results from fertile men. And this should be examined and better explored and published. And we could also have the choice of appropriate treatment of the man. When should it be full gonadotrophin treatment? When could it be, the tamoxifen or estrogen receptor blocking agents and so on? And these are always different, are most likely to be different limits. And I would say follow-up of treatment of male disorders. We know that men with a primary hypogonadism, secondary gonadism, who get gonadotrophins, they are quite likely to cause to start a pregnancy with their partner when they have extremely low values compared to when we look at infertile men. And I would say also the choice of appropriate assisted reproductive technology, it cannot be decided simply by the 5th percentile of the fertile men. We have to look more for when is ICSI really absolutely necessary, when can IVF function. There are, for instance, papers indicating that, or quite a number of studies indicating that if this progressive motility is very poor, then ordinary IVF won't work. So the percentage progressive sperm can be quite good, the number of sperm can be quite good, but it will not be useful to use the ordinary IVF. It will be necessary with ICSI. And I would say also very important, because the WHO manual actually was started to be a help to follow up the contraceptive intervention. The WHO worked to find a male contraception. And that is much lower. We have to find even few motile sperm to be able to say, well, that there are not a few motile sperm to be able to say that the contraception is efficient. And that actually goes also for vasectomy. So that was a way of pointing to something that is important in to develop more, to understand more of not only saying that we need one sperm for one egg and then we can make children, to understand more of the male reproductive organs, the male functions and disorders in the male, and actually look for treatments of that and see when can we use certain types of treatments. So, training of morphology assessment must be done properly, and it can be done properly. It has been, especially David Mortimer has been working on that, as well as the South African group, Roelof Menkveld, who have studied ways of training people to distinguish between low and high sperm morphology assessment. That should not be forgotten. Assessments of skills must be controlled regularly by internal quality control, and EQA is essential to evaluate information from other laboratories and thus necessary to ensure comparability between laboratories, and I would say also for the individual laboratory to use published data from other centers. And reference intervals from fertile men are not good for evaluating men in general. They are not good for the function, to evaluate the functions of the reproductive organs, and for selecting further investigations and treatment modality. We need more decision limits. So that was, yeah, that was the take-home message. Many thanks for your attention.
Lars, thank you so much. What a great and thought-provoking talk. I have got a question. I confess I'm asking this because it very much resonates with our own clinic as well, from Gulam Balador, saying, look, the problem with internal quality control is our clinic certainly has one guy that does almost an entirety of the semen analysis, and there's a few other laboratory embryology practitioners that then do the residual of that. And if you've got an internal control where you've got one person doing the majority of everything, should you not be trying to control to them rather than the middle ground? And similarly with an external program, would you not be better off to compare your frequent flyers, the people that do the majority between labs, rather than an average of each lab's quality or results performance?
I'm not sure that I understood all the questions there, but yes, if you have one person, you could come very far by using the archive data, so that you compare the fresh ones with the diluted or undiluted sperm. That's difficult, but you can have recordings of motility, you can have slides with morphology and vitality, and so on. So even if the lab is one person, they can do that, and they can complete it with participating in the external to know that the level is actually according to them. And what I've been involved in, actually, when I got the commitment from Lynn Fraser, David Mortimer, and Chris Barrett some 21 years ago, 22 years ago, was that it is essential for an EQA scheme to have target values not based on all participants, because there could be many new participants, new laboratories where they have not implemented quality control well. So therefore we implemented a scheme where we have 5 laboratories where people, not all participants there from each laboratory, but those who have been trained for quite a while, actually make a reference lab cohort.
There was something else that you just said, Sarah. Were you suggesting that your laboratory, everybody does the EQA material, then you send back an average?
No, what I was saying is, if you've got one practitioner or embryologist that's doing, say, 85% of the semen analysis, should you not use them as your comparator to another lab rather than using,
of course,
other people's data as well?
So if you're participating in EQA scheme, you have one person in your lab who's delegated to be the external comparator, and they do and submit their results to the EQA scheme. Then within your lab, you can use the EQA material as well as your IQC material to run your internal assessments, and that person is basically the anchor for that program then.
Yeah. I get you.
And by that person staying fixed against the external reference labs, you've now got this person can act as the anchor for the internal QC. And when, obviously, yes, if you've got one person doing the majority, that person's the poor bunny who has to do all the EQC stuff as well.
Yeah, that makes perfect sense. Now we've got.
If I may,
sorry,
we actually met this problem that there are laboratories who have had difficulties starting up with the internal. So therefore we have allowed, and now we have made it clear that we should not have averages from the laboratories, but we can actually, in the scheme that we are running from the Karolinska, we can take more results to help setting up the system for internal quality control.
Yeah, absolutely. And I mean, I think one of the other questions that's in the Q&A, and I would like to ask this to all three of you, is, you know, morphology is such a subjective thing. You're always going to have individual interpretation and individual error. And, you know, how do we cope with the uncertainty of that? Apart from obviously what you've said already, training, I'm sure that's what your answer will be, Lars, but can you elaborate a little bit more on that?
Yeah, well, as I explained in the beginning, we start with going through what has been published. And actually, when I've been very uncertain, when I started out this, and there were other, I have consulted people, and we have now a regular exchange with Roelof to be able to discuss what does this, well, it's easy, well, not easy, but it's quite comprehensive to write down criteria. But then when we sit there in front of, in the microscope, and that is why I said, when we have trained people in the laboratory, we have those common sessions. We train the novices not only by read this, but look at this sperm, and we discuss them. And when we train the, well, when David put us on training in the Stockholm laboratory, we started out with this also for motility, that technicians were sitting together, at least 2 and 2, looking at the same screen at the same time, counting the 4 categories of motility. And after a while, they agreed on it. And then we shifted because we had quite a number of lab staff at that time. So even, yes, there is in everything of this, there is part of subjectivity. And the point is then to address that. And as I said, if we have a member who, when we have the IQC and especially one slide where they are missing out, for instance, short tails, well, that is an easy, but then we go back and look at that. So that is the way to treat it. In the beginning, it's a little time consuming, but it's paying off quite rapidly still. And this with making that the Mortimer method with the graph is extremely good for the confidence of people, even those with little poor self-confidence and so on. When they see that actually the results, it's more difficult with numbers. It's better with the graph. Most people have easier to look at the graph.
David, did you want to come in?
It takes a good number of hours of actual one-on-one training with the novice, yeah. And then they go and read a series of slides, and you then, but because they've been scoring and keeping track of the different defect categories, you can go back and see what they're missing or what they're being too critical on, and you can then give remedial training, look at more sperm, discuss more things. And that graph that Lars showed, that was done back in the 1980s in Calgary, and there were 30 slides in each of those series. And the same slides may have been used in different series, but they were all coded and randomized. They didn't know what they were reading. So they were reading 30 slides per assessment. We use 20 these days, but 30 slides per assessment, 8 series. That's 240 slides were read before that person was judged competent. And we'd go back once a quarter and do the internal QC as well, with another few slides.
So next question then to you all again, if you've got any kind of comments on artificial intelligence or computer algorithms that can learn like humans, can those systems learn like humans, and could they have a utility to try and standardize morphology assessment in any way?
David, who or I?
Okay. First rule of AI is garbage in, garbage out. If you don't train those models using good data, they will not be useful, or they will not be able to perform as they should. So you can't train them against CASA machines, because virtually very few CASA machines can do full morphology. They look at heads. If you look in the mid-piece, but very few will tell you the length of the tail, for example, and you can get sperm with 35 micron tails or 40 micron tails, and they're short, and it's a defect that is often missed. So unless you've got real accurate data, you will not train properly. But I think, yes, it's going to be good, because the problem is that trying to use classical image analysis to do human sperm morphology assessments has proven to be extremely difficult. And it's partially due to the difficulty of imaging the cells unambiguously, but also to someone being able to translate what we see with our eyes into computer code. There was a guy who was really, really good at it, a guy called Damien Scouvert, who worked in Paris, and he had a fully automated sperm morphology system back in 1980, 81. Now, it was a Quantimet 720 at the front end, the PDP 1134 at the back end, took an entire room, and it ran overnight to read 30 slides. But it did a full French David classification system. So it can be done, but it was required someone with a great artistic talent to be able to translate what you see visually down a microscope into computer code. Damien is now retired and illustrates children's books.
So I hope that answers your question, Ralph Henkel, and I'm going to hand over to Kelly to put some more questions to the panel.
Yes, just one question again on the CASA system, because that's what a lot of people have questions about. And since we are now in the technological way of assessing sperm morphology, because it is indeed subjective. We prepare samples for our EQC here in Belgium, and we actually put the same sperm sample in two morphology slides, one in a high concentration and one in a low. And as you all can imagine, even expert labs scored the low concentration sperm lower in morphology than the higher. So there is a human subjectivity when we look at the sperm sample. So Bronte Stone has a question on the sensitivity of CASA sperm morphology algorithms. What do you as experts believe? Is there a future for CASA? Is the sensitivity good enough? If we want to count 1,000 spermatozoa to have a good morphology assessment, we are not going to do that manually. We're going to use a machine for that. So don't you think that in the future, we will be better off counting morphology with CASA systems? And then my following question is, why on earth is CASA still considered a research tool in the 6th edition of the WHO? Gentlemen.
If I might come in, I think that looking at the CASA system is not an eye, and it's not going to pick up things like vacuoles. It's not going to pick up certain abnormalities which you can pick up with the eye. So I think the eye is much more accurate than a CASA system. And coming back to the previous question, is I also use a scheme. I start from the other side. I look at the head, and I said, is it the head? Is it an oval form? And morphology is just, it's much more wider than just looking at the form. You must look at the size of the sperm. You must look at the size of the apple cell. You must look at the neck junction. You must look if there are cytoplasmic material present. You must look at the mid-piece, if the mitochondria has shifted, yes or no. There are so many factors that you must take into consideration that just looking using an artificial computer system is not going to give you the same information that you will be getting looking at the eye. The basic computer system is just going to give you the basic information and not the detailed information which you can obtain by looking at it yourself. And therefore it's important that you should know what is a normal size sperm. If you not know, then you measure that so that you also your stainings has different measurements, and these things must all taken into consideration. So sperm morphology is not something straightforward. That you have to keep all these factors which can influence the sperm. If you use testing, for instance.
Hey.
Hello.
Thank you, Roelof. Do you think maybe this one is for Lars, that a worldwide standardized training website would be the way to go for improving morphology analysis? A question from Philip Chan.
Yes, and we have been discussing a little, so to say, on the ESHRE basis, where ESHRE Special Interest Group in Andrology have been leading, and the possibility actually to set up courses, because we have started to experiment now in the Corona times to instead of, well, there are important aspects where people should be in the same room, but not that it works quite well, even over very long distances, to sit in actually a situation like this one, and with a microscope and moving around and looking at sperm and trying to discuss why is that sperm head not okay, like Roelof just described, looking at that. And yes, this tail is very short, but sometimes there are also technical problems. It is one thing what you can see yourself in the microscope, and what is the focal depth for the camera, what is the resolution of the camera. It doesn't seem nowadays that the bandwidth does so much problem to the system.
True. Yeah.
But I agree that would be, I would say, an ideal way of having international workshops where looking at and discussing. I think that is more important than, so to say, have a number of photos just posted on the website.
Yes, absolutely. So SIG Andrology can contact SIG Quality and Safety, and we will team up and make it happen for sure. One last question from me to David in the chat, and then I'll give it back to Sarah. Whatever happened to IMSI?
I have no idea. I never saw a really need for it, because I have no idea
What
where
do you
those
think
patients
was the
came
main
from.
flaw?
It's very expensive. It takes a huge long time. But as far as I know, every Brazilian clinic does IMSI on almost every patient, from what I'm told. So it's there, but I just don't think it's quite as upfront as it used to be. But I think IMSI per se is a combination of two things: the Sperm morphology method and ICSI. And I think the key thing with IMSI was that using slightly better optics to be able to avoid those sperm with the craters. And I think the problem here is that if people aren't trained to differentiate craters and vacuoles, or what do you want to call those, nuclear inclusions, wherever, people aren't going to know what they're looking at. I have seen presentations at conferences where people point to both things and call them all vacuoles because they've never been educated about them. So this is why training is important. And I think that these things will be easy. For me, I think any sperm that has a crater should be considered abnormal because it is associated with evidence of dysfunction. I don't think there's the same evidence for vacuoles. So perhaps that problem that Rolf was saying where you can't find the evidence is because people weren't scoring vacuoles different to craters. So the question wasn't asked correctly because the data wasn't there. And I think this is partly training, it's partly for imaging quality. CASA can do lots of things, but it has to be programmed. The imaging has to be good enough. Early days, it wasn't up to it. I think the most recent systems have that potential, but they have to have the investment in improving their image analysis capabilities. There have to be better algorithms. But I think that for a lot of the subtleties we're looking at, two things that's important. One is we really need to have a standardized method for preparing sperm morphology smears and standardized stain and fix them and stain them and mount them. So you've got a proper reference material. If we still use 16 different types of stains and mount them, don't mount them, whatever, it's not surprising people are going to get different perceptions. But we have to have that improved. But we also, I think AI is the way of the future, because it's going to be able to, it will be able to look at things the way we do if we present them to it with the classifications. So I think that is the huge potential, because right now, car systems, for most of them, don't even look at the tail. Or they go, tick, it has a tail. That's about as far as it goes. So it's not going to get us there. The other thing is
so maybe for the 7th WHO, right? It will be there.
God knows.
Not as a research tool.
The one thing I want to point out is that without using this sort of technology, we will never be able to get places to read enough sperm. I mean, if you are just thinking about what happens, if you count 200 sperm and you say this man's got 4% normal forms, actually it could be 2, 3, 4, 5, 6, 7, or 8%. That's all within the statistical sampling range of measurement error. So yes, on a population basis, you can say there's a difference at 4% between good and bad prognosis. But for an individual man with a huge measurement error based on counting small numbers of sperm, you don't even know whether he is 4% or 3% or 5%, 2% or 7%. So you're just misleading yourself. So we need to be able to analyze hundreds, preferably 1,500, 2,000 sperm per slide. Nobody counts 2,000 sperm per slide, except I did in my PhD, because that was how I needed solid data. But I wasn't trying to do it as part of a semen analysis. It was a research study. Yes. You can do lots of things in research you don't do in routine. But if you do that, you will get useful information. But I don't see it happening in routine labs. It's going to have to come with computer technology. And I think it's going to be AI that's going to take us forward. And the other part of what the WHO says about CASA, I believe is until the software is something that has like FDA certification or whatever, it can't be considered a clinical tool because it's not allowed to be used as such without various regulatory approvals as a medical software.
My embryologists are not, don't have a label on them. Come on.
No.
Sarah, please help us out. Have you found any questions in the Q&A?
Well, please, may I just, Kelly, you asked me about why the WHO has not put CASA as a routine tool.
Well, you stated it as a research tool.
Yeah, and that is because the lack of sufficient scientific evidence. We are seeing that there are things going on, but yes, as you said, maybe the 7th or 8th. But at present, we can't find that there is evidence enough for the usefulness to recommend it. Yep, that's all.
If I must just come in again, the strict criteria is not looking strict at the sperm head as such. The strict criteria is a holistic approach that you do the best you can to get the best smears, the best staining, apply what is described as the best methods available. And looking at the sperm head is only part of the whole process of sperm morphology evaluation by stricter application of the guidelines that are available, and not making poor smears, making poor staining. It's a holistic approach, and looking at the head alone is only a very small part of the whole strict approach to sperm morphology evaluation. If you don't do the basics correctly, you won't get a good sperm morphology result.
If you make poor smears and you stain them with some horrible stain and make everything different blobs of purple, don't expect to get anything meaningful out of it.
Yeah, if you see the literature, I mean, photos and in journals, with the kind of staining they have, you can hardly see that's a sperm, and then they try to make morphology evaluation and make conclusions, which is just impossible to do.
I still come across embryology labs where they try and do their morphology assessment on swimming sperm in the macula chamber.
Yeah.
I wish I could do that. I've never have been able to do it. And I've been doing it since the mid-70s.
Assuming that you have prepared your slides appropriately for morphology and you have a patient that has 0% morphology, 0% normal morphology, what are you going to do with that man?
Personally, if he has a low pterosaur sperm index, we could still, and his sperm will hyperactivate, we could do IVF.
Yeah, it depends also why he has 0% morphology. If you have a patient with globospermia, of course you're not going to get the results.
Yeah.
But if you have patients with mainly slightly elongated spermatozoa, those are not pathological, and you can still expect good results with them. So that's why I said it's not just good enough to say you have 4% or 0% normal morphology. You must specify what you see and why you make that that conclusion. So saying just 4% is not good enough.
You need to know. I mean, that's why we use the TZI. We want to know how abnormal those abnormal sperm are. As Rolf says, they just got slightly fin-shaped heads. It's probably utterly irrelevant. And
so or as a clinician, I get a report and I don't get all of this insider information that you all know about. I get a report saying 0% normal morphology.
Then ask them for the various morphology defect categories and a TZI result as well.
Yeah, we do TZI.
Yeah,
so you can have a normal head, but you can have a shift of your mitochondria. So the sperm is not going to have the correct movement. So
hyperactivation is a key thing as well. So the thing is that actual sperm head shape is heritable. Some men do have slightly more oval shapes. Some have slightly more rounded shapes. I mean, so a whole lot of sperm morphometrics are actually heritable characteristics, and that's not just human. That's in all mammals. So, and you can actually breed in mice, for example. You can breed for longer mid pieces. So, you know, you've got to understand that some of this is a little bit of auto recalibration to the man as well.
Okay. Yes. Thank you. Thank you very much. Unfortunately, we are running late, so we will have to wrap up and close this event. And by the number of people, attendees this evening, registrants and questions that we have not been able to go through all of them, I think it's very, very important that we get those basic information on sperm morphology and physiology back on the table, because I think there is a need for that. So hopefully we will see all of you again in some really good courses. And I think indeed ESHRE and other scientific organizations can help by spreading the information and the news so that we all get better at analyzing not only morphology, but also other items about sperm. So thank you everybody for taking the time to participate in this session and the discussions. I particularly would like to thank the I3 sponsors Zymote and the I3 speakers for this evening, David, Roelof, and Lars. Thank you very much for your expertise and your discussions. Thank you to the organizing committee of this initiative, Jacques Cohen, Peter Nagy, Giles Palmer, Thomas Elliott, Shaista Sadruddin, and Marianne Svetes. And today's Q&A team were Shaista, Marianne, and Anna McLaughlin. Sarah.
Thank you, Kelly. So next week is another session, same time, same place. And it's the third episode in a trilogy, Cryogovernance 3, Thin Ice. And your moderators will be Dr. Alison Bartolucci and Professor Catherine Rakowski. And you've got speakers in the form of Dr. Victoria Santos, who's talking about too hot to handle, determining devitrification thresholds. FMEA IT, a qualitative and systemic tool for cryogovernance by Dr. Kirsten Ivani, and Clean Dewars and Glass Houses by Dr. Martine Nies. So goodbye from all of us for now, and thank you for joining us.
Thank you. Good night.
Everyone.
Thank you.